Welcome to the adaptive immune system. To start, let's introduce you to the special cells that make up the adaptive immune system. These consist of helper T-cells, cytotoxic T-cells, and B-cells. These cells will interact with two other different types of cells in our bodies, which are your average run-of-the-mill body cell, as well as a special type of cell called an antigen-presenting cell. Every single nucleated cell in our bodies, so every single cell except red blood cells, has something on them called an MHC class I molecule. The MHC class I is often called the window of the soul. This is because all of our cells are constantly taking samples of everything within their cytoplasm and displaying these samples in the MHC class I molecule for the world to see. However, antigen-presenting cells also have another specialized molecule called an MHC class II, which allows them to present antigen that they've processed to our helper T cells. The three cells that make up this group of antigen-presenting cells are macrophages, dendritic cells, and B cells. When an antigen-presenting cell, like a dendritic cell, comes into contact with a pathogen, it will perform phagocytosis to take in the pathogen, to kill it, and to break it into tiny little pieces called antigen. It will then put these tiny antigens into its MHC class II molecule and go looking for our first adaptive immune cell, the helper T cell. The dendritic cell will then come into contact with every helper T cell that it finds, performing sort of a handshake with their T cell receptors, looking for the T cell whose receptor is specific for that specific antigen and is able to bind to it. Once this happens, the helper T cell will then strengthen this binding with a molecule called CD4. After this, the antigen-presenting cell will express co-stimulatory molecules called CD80 and CD86, which will then be bound by CD28 on the T helper cell, giving the T helper cell the second signal. Finally, once this has happened, the antigen-presenting cell will release what are called cytokines that will bind to the cytokine receptor on the T helper cell, giving it the third and final signal, thus activating this T helper cell. This active helper T cell will then rapidly divide and differentiate into many different types of helper T cells with different functions. It will also form memory T cells. The memory cells will stay in the secondary lymphoid organs, like lymph nodes, waiting patiently for a repeat infection with this same pathogen so that they can be ready and act much faster and more aggressively, because these memory cells will only need the initial signal of binding to the antigen to once again become active. These activated T helper cells that aren't memory cells are referred to as plasma cells. The plasma cells will immediately get to work, performing whatever task they were told to do by the antigen-presenting cell, be it activating B cells, supporting the other immune cells at the site of infection, or activating other immune cells needed for different types of infections. As I just mentioned, some of these helper T cells, that we call TFH cells, or T follicular helper cells, will go out activating B cells, so that the B cells can help fight the infection. In order for this to happen, a B cell receptor on the membrane of the B cell needs to come into contact with the specific antigen that it codes for. Once this occurs, the B cell will perform endocytosis to bring the antigen inside the cell so it can then break down the antigen and present it on its MHC class II molecule, just like the dendritic cell. Once the B cell is showing antigen, it looks for the TFH cell that was activated by that same antigen so that it can come bind to it. The TFH cell will bind to the antigen using its TCR and strengthen the binding with CD4. Once this has happened, the B cell will express its co-stimulatory molecule called CD40, which is bound to by CD40 ligand on the TFH cell. Following this, the third and final signal is given by the TFH cell releasing cytokines that are then bound by the cytokine receptors on the B cell. Once the B cell has been activated, it will divide rapidly and differentiate into plasma cells as well as memory cells. The memory cells will wait, just like the memory helper T cells, for the next infection with this pathogen, ready to strike at a moment's notice, only requiring the initial binding to antigen to be activated again. The plasma cells will immediately begin secreting antibodies, then move to the bone marrow, where they just continue secreting antibodies to help fight off the infection. Different types of antibodies have different functions, which include, but are not limited to, stimulating an allergic reaction, binding to the antigen and grouping pathogens together so that they can't bind to your cells, and enhancing phagocytosis. Next up is the cytotoxic T cell. In order to activate a cytotoxic T cell, an antigen-presenting cell will perform phagocytosis on a virus. Then, after getting special permission from a helper T cell, it will show this viral antigen on its MHC class I molecule. The antigen-presenting cell will then search for exactly the right cytotoxic T cell, who's got the T cell receptor that is specific for that viral antigen. It will, just like with the helper T cells, shake hands with every single cytotoxic T cell that it comes across until it finds exactly the right one. This cytotoxic T cell will then bind to the antigen using its T cell receptor and strengthening that bond with CD8. Following this, the dendritic cell will express its co-stimulatory molecules CD80 and 86 to the cytotoxic T cell, which will be bound by CD28 on the cytotoxic T cell. For the third and final signal, the dendritic cell will release cytokines that will bind to cytokine receptors on the cytotoxic T cell, thus giving it the final signal to say, yes, it is okay, you can go out and perform your function. The cytotoxic T cell will then rapidly divide and differentiate into memory cytotoxic T cells and plasma cytotoxic T cells. The memory cytotoxic T cells will sit and wait for the next infection with that pathogen, being ready just like the others, and only needing that initial binding to the antigen to be activated. The three-step activation process is extremely important, especially for cytotoxic T cells, because their job is to literally go out and kill any cells of yours that have been infected with an intracellular pathogen like a virus. Let's show how this is done. First, a body cell will come into contact with a virus because the virus attaches to the body cell and then injects its genome within the cytoplasmic membrane and into the cytoplasm of the cell. Following this, because the cell is constantly sampling all of the proteins that are in the cytosol and presenting them on their MHC class I, this includes any viral proteins that are being created by the hijacked machinery that the virus has taken over. So it will take these small pieces of viral proteins, package them up with the rest of the proteins within the cell, and present them on the MHC class I molecule. Meanwhile, an active cytotoxic T cell will be patrolling the blood, looking for any cells that have irregularities in their MHC class I. When it finds one, like this body cell that's been infected with the virus that it's looking for, it will bind to the antigen that is in the MHC class I using its T cell receptor, following which it will strengthen the binding with CD8. Once this has happened, and the cytotoxic T cell recognizes the virus within that cell's cytoplasm, it will release cytokines called perforin and granzyme. The function of perforin and granzyme is to punch holes in the cytoplasmic membrane of these cells and induce apoptosis, thus killing the cell and rendering that cell useless and helping the virus to continue to replicate.